Hello, everyone. This is Catherine Ragimbeau from Luxembourg. Thank you for inviting me to this Campylobacter event, especially in memory of Alison Cody. She inspired me so much for my research work on Campylobacter. I had the opportunity to work with her over the screen, I would say. In 2012, she proposed me to contribute to the creation of the POR-A database in PubMLST. And that was a very good surprise, and I accepted immediately. And we had a very nice collaboration together. And of course, we shared the same scientific interest and the same favorite bacteria. Today, I will present an investigation about the phenomenon of recurring genotypes of Campylobacter jejuni in human infection. And I will give some perspectives on in monitoring Campylobacteriosis. This work was done in a framework of a research project named Campylomic, in collaboration with different partners, as you can see on the bottom of this slide. And the project was supported by Fonds National de la Recherche in Luxembourg. So let's start by the background of this study. We have a large collection of isolates in Luxembourg, over 15 years now. Actually, we receive each year around 600 Campylobacter jejuni from hospitals and medical labs. And but we also have animals and food isolates from the veterinary lab, and also a collection of isolates from environmental samples through different research projects. And the first one was launched in 2005, and the last one is still ongoing. So at the end, we have one database with human and non-human isolates, and we do molecular typing. So a long time ago, now we have implemented an in-house molecular typing system. Actually, it's an extended immunity analysis. We have combined the immunity scheme from Oxford with two other loci under selective pressure. The first one is a subunit A from the gyrace, which is interesting to know, to have the information if the strain is resistant to quinolone. And there is also a kind of host signature and associated with poultry. The second locus is, of course, POR-A, which is a very polymorphic locus. And for us, a definition of genotype is a combination of EST, G-rays, and POR-A alleles. And with these methods, as you can see here, we have quite good discriminative power. With this quite high discriminative power, we were able to detect human cluster. And this is an example of, I give you 4 examples of genotypes. And each square, you have the definition of the genotypes here. And on this axis, you have the number of human cases, and the timeline is starting for 2006 till 2019. As you can see in this first square, this is a timeline distribution of human cases infected with this genotype. And the pattern is quite endemic for this one. And we also have identified this genotype in poultry in bovine, and in surface waters. For the other, have a microepidemic pattern for these two. And this one is quite emergent one. And this one has a strong epidemic curve. This is the most important we had in Luxembourg. It was in 2014, and around 70 patients were infected with these genotypes. What is important to know, it's each year we have around 50% of the human cases associated with recurrent genotypes. So the question was, but do we have the same classification of the isolates if we use whole genome sequencing? And so for this study, we have selected a panel of strain, and as you can see, it's 4 lineages that I have presented you just before, and from different sources. And we had a collection of isolates with unique genotypes. And then we did the whole genome sequencing by using the Illumina technology. And then we had set up quality control criteria. We compared only data with no more than 2% of missing target for each typing schemes we have tested. And then we had the classification, of course, but the gene by gene approach. And here, this is a brief presentation of the pipelines that we have tested. So we have the core genome MST developed by Ragimbeau Sexfer, the one from Innuendo, and the one from Oxford. It was the one established by Ali. As you can see here, the number of targets is different. And here you have the cluster alert distance. This is a rules that allow you to group together here. And for Sexfer, it's into a complex type, the similar genomes. Here the rules is the cutoff is no more than 13 allele of differences. And for Innuendo, the cutoff is of 4. And for Oxford, there was no cutoff. And so we defined one by dynamic core analysis. And this is the results. And to visualize the results, these are minimum spanning trees for each method. And you have Ragimbeau Sexfer here, Oxford, and Innuendo. And each color represents strains associated with one lineage. And in gray, you have the unique genotypes. So there was, as you can see, for example, lineage in blue is the same. It's quite the same in each of the methods. There is a good concordance in between the 3 core genome MLST tested, with a few differences. Actually, I would say that the Oxford and Innuendo have a little bit more discriminative power than the Sexfer one. You can see it, for example, with unique genotypes. Here you have 2 isolates group in the same complex type, and this is not the case with the other typing scheme. These results were recently published by my PhD student, Morgan Nenin. And what is interesting is that raw reads are available by ENA. And one country has already contacted me by saying, hey, I have a lineage B and lineage D in my database too. And this is very important for the surveillance. About surveillance, I would like to present a little event that took place in 2020 on the AP Food and Waterborne Disease platform, which is a platform for surveillance hosted by ECDC. In 2020 was the first year with 2 urgent inquiries, including genomic data. So this is very important for the surveillance, and it was 2 countries, and in both cases it was related. And so we did some comparison, and Luxembourg replied. And I will give you some results. For the first urgent inquiries from October 2020, the definition of the genotype was ST19-1, which is fully susceptible to quinolone, and POR-A7. Yeah, I will say that I must say that even if I compare strain by using core genome analysis, I'm still using the nomenclature ST, GRAS, and PORIN, as you can see. And this combination is almost the same than the one from lineage A that I presented before, corresponding to the endemic one in Luxembourg, except that our strains are resistant to quinolone. But still, we have some isolates that clustering with these reference strains from ruminant, human, and chicken. And when we do the comparison, if you have a look at the difference matrix, you can see that the difference is very low, and even 7 allele of difference with a chicken sample from 2011. So it means that there is a strong stability of the core genome of this lineage in space and time. And for the second urgent inquiries launched in November 2020, the definition was ST7355, associated with GRAS9, which is resistant and has a signature for poultry, and a new allele of PORIN. Yes, 2020 was not the best year for the surveillance of Campylobacter in Luxembourg. We received only 237 isolates from human. And this is a distribution of human cases over the year, starting from December 2019 to December 2020. And if we look at recurring lineages, genotypes, we had in 2020 17 recurring genotypes, and 40% of patients were infected, were associated with these recurring genotypes. And what about this specific genotype from the UI? I found 13 cases linked, and this is a distribution of these cases. There is no peak. We can find some cases at the start at the beginning of the year until September, October. So, in summary, whole genome surveillance support the presence of recurring genotypes in human infection. And we have these different kind of patterns, endemic or epidemic, or micro-epidemic. There was a good agreement between the 3 core genome MLSTs that we have tested, but the results also suggest that isolates sharing the same ST, G-rays and PO-A combination of alleles also share the same core genome. I didn't show you, but even with the whole genome MLST, we did some comparison, and the result supports the presence of stable genomes. And these stable genomes represent a risk factor of geographic spread that needs to be investigated further. At the end, I would say that the Campylobacteriosis is not only of sporadic nature. There is a strong interest in monitoring genotypes involved in human infection. Thank you for listening, and thank you to all the detectives who took part in this work. And welcome for your questions.